Welcome, and thank you for joining me today to hear about a specialized whole food fermentate. I'm Justin Green, PhD in biochemistry, and I'll be giving you a quick overview about fermentate's background and then the science behind fermentate's beneficial effects on the immune and gastrointestinal systems. First, I will discuss in more detail what I mean by whole food fermentate and what makes it specialized. Safety and quality will also be discussed, which are of utmost priority in manufacturing fermentate. Second, I will get into the meat of the presentation, the science that has been done that shows that this specialized fermentate provides more healthy days, is fast-acting, and overall works to strengthen and rebalance a healthy immune system. Lastly, I will introduce new science that links the benefits that fermentate gives to immunity and to the health of the gut. So now let's look at what makes fermentate a whole food fermentate. Fermentate is made with a yeast commonly known as baker's yeast, or scientifically as Saccharomyces cerevisiae. Like fermentate, many foods are made from a fermentation process that results in a whole food product made with yeast. For example, yeast metabolism causes bread to rise through the release of carbon dioxide. Also, yeast metabolism produces alcohol and flavor in beer, wine, and liquor. Finally, yeast fermentation produces the unique nutrients and flavor found in a multitude of other foods, such as Vegemite. In all these examples, the end product is completely different from being simply yeast. It's the food that's made with the help of yeast fermentation. So how is this fermentate made in a specialized manner? First, the yeast is fed a specific set of nutrients. This blend has been honed over 70 years of fermentation experience. Second, the fermentation cycles in a specific manner between aerobic and anaerobic processes. In other words, with or without oxygen. Yeast produces different factors depending on whether it's growing with oxygen or without oxygen. So controlling these processes ensures a wide but consistent range of metabolites. Then the fermentation solution, which contains not just the yeast but what the yeast has produced, is dried in a specific manner. As the yeast evaporates, the yeast dies or is inactivated. However, the heating process is gentle enough to cause limited degradation to the important metabolites that the yeast produced, such as polyphenols, polysaccharides such as beta-glucans, proteins, peptides, antioxidants, minerals, and others. This process produces a complex mixture of ingredients, and it's thought that this complexity is part of the basis for its beneficial activity for the gut and immune system. However, this complexity means that consistency must be monitored closely. One of the many quality tests that is used to ensure this is called Fourier transform near-infrared spectroscopy. It is essentially a way to provide a fingerprint for this mixture, and using it ensures consistency every time the specialized fermentate is produced. The safety and quality of the specialized fermentate is of absolute priority. A battery of safety studies has been published in the International Journal of Toxicology, and the United States Food and Drug Administration has looked at the fermentate studies and manufacturing processes and has acknowledged that fermentate has new dietary ingredient status. Good manufacturing practices in the production of fermentate are regularly certified by NSF International through frequent audits. There is a HACCP plan in place. HACCP stands for Hazard Analysis Critical Control Points and is a system concerned with ensuring ingredient safety. Fermentate has GRAS status, or generally recognized as safe status, in a wide variety of food applications and is considered natural, vegan, non-GMO, kosher, and halal. It is currently incorporated in a wide variety of supplement products throughout the world in various delivery forms such as capsules, tablets, chewable tablets, soft chews, stick packs, effervescent tablets, liquids, powders, and liquid shots. Note, in case you're wondering, common yeast infections are not due to baker's yeast, but from another yeast species called Candida albicans. Next is the science that has been performed specifically on this specialized fermentate. The depth and breadth of these studies distinguishes fermentate as having one of the most extensive evidences of modes of action, along with demonstrated human and health benefits in comparison to any other immune nutrient available. Up to 2016, there were 7 human clinical trials that have been published on fermentate, including multiple double-blind placebo-controlled randomized trials, or RCTs, using over 300 subjects. RC human clinical trials are the gold standard for clinical studies, and they are recognized as the strongest form of substantiation for desired benefits. Furthermore, it's important to note that healthy, non-stressed populations were used in the studies on Fermentate, just like the people who would be taking this supplement day to day. These human trials are what I'm going to mainly talk about today. The health endpoints they cover include incidence and duration of cold and flu symptoms, the incidence and duration of allergy symptoms, and the rapid response to supplementation as measured by immune-related biomarkers. They are backed up by a series of in vitro and model studies that delve into inflammation, mechanism of action, and biomarkers. 2017 will see the publishing of an 8th human trial on gut health and the microbiome, and I will conclude this presentation talking about this link between immunity and the gut health. First, I will discuss 2 studies that looked at the incidence and duration of cold and flu symptoms. These studies are instrumental in backing up Fermentate's role in immune health because they use a model system that specifically correlates to human immune function, and they were done on healthy, non-stressed population. The first study, trial number 1, was a randomized, double-blind, placebo-controlled human clinical trial. It was performed over 12 weeks during the major cold and flu season, in the middle of winter in a U.S. state, South Dakota, which is known for very cold winters. 116 healthy subjects participated in this study, and the subjects were chosen to be non-vaccinated for that season's influenza virus. Thus, these people, while healthy, were especially at risk for acquiring the influenza virus. The non-placebo group took 500 milligrams Fermentate once daily. 500 milligram daily is the standard dose for Fermentate and has been used for every human clinical trial thus far using this specialized Fermentate. The incidence of symptoms that correlate to colds and flu were recorded for all participants, and it was found that the group taking Fermentate had a reduced incidence of cold and flu symptoms compared to placebo group. This improvement was statistically significant as evidenced by the low p-value of 0.01. The second study was conducted concurrently by the same research group. It was also a randomized double-blind placebo-controlled human clinical trial and performed over the same 12 weeks during the major cold and flu season in South Dakota. However, for this study, the 116 healthy subjects were vaccinated for that season's influenza virus. Again, the non-placebo group took 500 milligrams Fermentate once daily. For this study, not only was the incidence of cold and flu for the Fermentate group lower compared to the placebo group, but the symptoms they did have had a reduced duration. In both cases, the improvements were statistically significant as evidenced by the low p-values. So not only are you seeing the incidence of cold and flu lowered with Fermentate, you're seeing the length of time when those symptoms occurred shorter for the Fermentate group versus placebo. Because both these studies were conducted concurrently by the same group with identical protocols, the data from both studies can be combined. The data shows that over the 12-week study period, there was a 26% reduction in the duration of cold and flu symptoms for the Fermentate group compared to the placebo group. Thus, the subjects taking daily Fermentate, combining the people who were vaccinated for flu and the people who were not, had more healthy days during cold and flu season than those who took placebo. Living more healthy days is one of the main reasons people take an immune health supplement, and the science shows that this specialized Fermentate delivered this result. Another benefit that immune supplement users want is rapid response. In other words, when you're about to get on a plane, or your kids are about to start school, or you're doing something else that might stress your immune system, you want to make sure that when you're taking an immune supplement that it works quickly. For the next study, I'll present evidence showing that this specialized Fermentate starts working within 2 hours. This rapid response clinical trial was also a randomized double-blind placebo-controlled human clinical trial, and its subjects again were healthy, non-stressed adults. Because the study was looking at rapid response, it could be conducted over a shorter period of time. The previous studies were looking at cold and flu symptoms and as the primary endpoint, and by necessity had to run over months. This rapid response study was looking for effects over 2 hours of time, and this shorter time frame enabled the researchers to give this study a crossover design. What this means is that all subjects were tested after taking Fermentate, again 500 milligrams, and those same subjects on a different occasion were tested for taking placebo. So each subject was their own control. There was a 2-week washout period between the 2 treatments. For each subject, blood was taken before treatment at hour 0 and 1 hour and at 2 hours after each treatment. The serum was then analyzed for immune activation. The next few slides will get somewhat technical. They will discuss the function of T cells and natural killer cells, 2 important immune health lymphocytes, or white blood cells, and how they first localize the sites of potential infections and then communicate with each other so that the fight against infections can be coordinated. It is technical. However, the main thing to take away from this research is that validated markers for immune activation were seen in greater effect after the fermentation ingestion versus placebo in just 2 hours. First, let's look at the results for T and natural killer cell serum concentration. T cells and natural killer cells are important immune health lymphocytes, or white blood cells. They are represented in this picture by the gray circles in a blood vessel. 2 of the jobs of white blood cells upon indication of infection is first to localize to tissues such that they start fighting the infection, and second, to communicate with other cells such that the fight can be coordinated. Lower serum concentration indicates tissue localization rates of these immune defense lymphocytes has increased. The reduction in serum concentration, in other words, the presence in the circulatory system for these important immune cells, does not mean that they disappeared. It means that they left the serum in preparation for an immune response. The results were that, compared to placebo, a transient reduction in serum concentration was seen 2 hours after fermentate treatment. Again, the reduction in the number of these cells in the serum indicates an activation of these cells had occurred. This reduction was statistically significant as indicated by these low p-values. Immunologists describe such a transient serum reduction of these cells as an indicator of tissue homing or healthy immune surveillance. What it means for those taking fermentate is that an important part of the immune system has been activated and primed for action, and that this activation occurred within 2 hours after fermentation treatment. One of the roles for natural killer cells in the immune response is communication. One important way cells communicate is by expressing proteins and displaying them on the surface of other cells for other cells to chemically see. These cell surface receptors are like flags in a war, telling the immune response cells what the state of an infection is and what the required course of action is. One such receptor is CD25. It communicates to other immune response cells to increase proliferation, or increase the number of immune soldiers in the war. Another such receptor is CD69. It communicates to other immune response cells to increase cytotoxicity, to get the soldiers' weapons ready. But for our purpose, what is important is that expression of these 2 receptors indicates that natural killer cells have been activated to get ready for an immune response. The results of this study showed that indeed, expression of these receptors was higher when subjects were treated with fermentate compared when they were treated with placebo. This activation occurred at 2 hours for CD25 and at 1 hour for CD69. The third measure looked at was antioxidant power. This was done using a method called CAP-E. Essentially, it is a measure of how well the serum itself can protect red blood cells from oxidative damage. Again, within 2 hours, fermentate treatment provided the serum with more antioxidant power than placebo treatment with statistical significance. This is important because the body suffers from increased oxidative stress when the infection is being fought. So in these 3 ways, it was shown that within 2 hours, fermentate ingestion activates the immune system within 2 hours. So, the studies I've presented so far show that fermentate treatments can give subjects more healthy days during cold and flu season, and that fermentate treatment seems to activate certain immune strength parameters within 2 hours. However, a wholesale boost to the entire immune system would not be beneficial. Sure, boosting all immune responses would help with infections, but it would also boost the response to allergens and other normally benign antigens, and this could lead to increased allergic reactions and other forms of unwanted inflammation. Allergies and inflammation are, in fact, a product of an overactive immune system. What is truly needed is a balanced immune system. A balanced immune system will respond well to general infections, but will not increase the immune response to harmless antigens such as pollen. How this balance can be achieved is complicated and is still being studied. For the purpose of this presentation, I will simply look at how treatment with this specialized fermentate affects the inflammatory response. As I mentioned earlier, there are fermentate studies using models of inflammation. They showed that fermentate actually dampens the inflammation response compared to placebo. With those results in hand, a human clinical trial was done on a specific but pervasive form of unwanted inflammation: seasonal allergies. Again, like the 2 cold and flu trials, this trial was a randomized placebo-controlled human clinical trial. It was done over 12 weeks during allergy season, and pollen count was monitored at this time, and it was found that the first 6 weeks of the study had significantly higher pollen counts than the remaining time. The results that were reported covered these 6 high pollen weeks. This is important because it indicates not only that fermentate acts relatively quickly, but that the symptoms seen during this study can be concluded to be due to allergic reactions as opposed to other reasons, such as illness. Moreover, unlike many clinical trials on allergic response, the 96 healthy subjects in this fermentate study were confirmed to be allergy sufferers by skin prick tests prior to inclusion into the study. During the study, each subject filled out a rhinoconjunctivitis quality of life questionnaire, which is a standardized validated method to monitor allergy symptoms. Again, subjects took a daily dose of 500 milligrams fermentate and were compared to subjects taking placebo. The clinical results showed that compared to the placebo group, the fermentate group showed reduced severity of nasal congestions and reduced severity of rhinorrhea, or runny nose. The fermentate group also showed reduced use of medications used to treat allergy symptoms, such as pharmaceuticals and inhalers. These positive results were all statistically significant, as shown by the low p-values. When you look at the number of days people suffered with nasal congestion, you see that the placebo group was congested for over 4 weeks of the 6 high pollen weeks of this study, so 2/3 of the time. The fermentate group, in contrast, had 43% fewer days of nasal congestion, or about 2 weeks out of the 6. This is a sign of significant relief for these allergy sufferers, and again shows that this specialized fermentate results in more healthy days. To conclude this presentation, I want to present some exciting new fermentate science on gut health. At first blush, the immune system and the gastrointestinal system, or GI system, may seem unrelated. But in fact, the GI system plays a central role in immune system homeostasis. It is the main route of contact with the external environment and is overloaded every day with external stimuli, sometimes dangerous factors, such as pathogens, like bacteria, protozoa, fungi, viruses, or toxic substances. In other cases, very useful factors, such as food and good bacteria, that is essential for functions such as fiber digestion. Your GI tract must serve as a very discerning gatekeeper. Keep the bad things out and defend against them, and let the nutrients in and allow the good bacteria to thrive. The crucial immune role of the GI system is testified by the huge amount of immune cells that reside within it. Indeed, gut-associated lymphoid tissue, or GALT, is the prominent part of the mucosal-associated lymphoid tissue, or MALT, and represents almost 70% of the entire immune system. Moreover, about 80% of the plasma cells, mainly immunoglobulin A, or IgA-bearing cells, reside in the GALT. I want to take a moment to talk specifically about that molecule, Secretory immunoglobulin A, or SIgA, because not only does it provide a link between immune and gut function, but it is also one of the factors stimulated by this specialized fermentate, and thus may be a clue into how fermentate helps both immunity and the gut. It is a specific form of antibody, a type of molecule that is the vanguard in fighting infections, that, as I said previously, is produced extensively throughout the GI tract, about 3 to 5 grams per day. It is a linchpin of the mucosal immune function, promoting the clearance of pathogenic organisms, quenching bacterial virulence factors, and thus influencing gut microbiota composition. It also downregulates some pro-inflammatory responses. If you remember previously, this seems to mirror fermentate's function: increased immune response against infections while dampening the inflammation response. And, in fact, fermentate treatment results in an increase in salivary SIgA. Human clinical results show that compared to a placebo group, the fermentate treated group showed an increased SIgA compared to placebo with statistical significance after 2 months, as shown by the low p-values. Because SIgA is such an important immune factor in the GI tract, these results prompted Fermentate research specific to the gut. The main model system that has been used to study Fermentate in gut health is called the Simulator of Human Intestinal Microbial Ecosystem, or SHIME. In this system, large beakers are used to simulate the different sections of the gut: the stomach, small intestine, and the 3 sections of the large intestine, the ascending, transverse, and descending sections of the colon. A sample of human gut microbiota, the bacteria that takes up residence in our colon, is introduced into the beakers representing the colon. Solution corresponding to pancreatic juice is introduced into the small intestine beaker, and solutions corresponding to the stomach, acids, and food, and perhaps a treatment such as Fermentate, are introduced into the stomach beaker. The beakers are kept at body temperature and undergo stirring. Solution is slowly transferred from the upper part of the model, the left side of this figure, to the lower to simulate the path of digestion. Digestive products and, importantly, the nature of microbiota can be monitored in each gut section over multiple time points. 2 important components of the microbiota are bifidobacteria and lactobacilli. These are generally considered good bacteria for gut health and are especially important for proper digestion. In the SHIME model system, Fermentate treatment was compared to treatment by 2 frequently used prebiotics, inulin and fructooligosaccharides, or FOS. Prebiotics are components, usually carbohydrates, that are normally ingestible by humans, but that nourish or help to stimulate the growth of gut bacteria. Well, the results showed that an increase over time of bifidobacteria and lactobacilli during treatment by various factors through the SHIME model system. The effect of Fermentate was on par with inulin and FOS, and does much better than simply cellulose, which is a negative control. Thus, it can be said that Fermentate is displaying prebiotic effects in this model system. This is especially striking because it is known that Fermentate does not cause the unpleasant side effects, such as intestinal distress, that can be seen for inulin or FOS treatment in humans. In the SHIME model, one can also look at digestive products. An important class of digestive products are short-chain fatty acids, or SCFAs. One SCFA, butyrate, is of particular interest because high butyrate concentrations in large intestine support a healthy intestinal epithelium and help control inflammatory and malignant diseases of the intestine. If you look at this chart, cell stands for cellulose, the control. FERM stands for fermentate, IN stands for inulin, and FOS stands for FOS. And as you can see, Epicor treatment was very good at increasing butyrate concentrations, beating each of the other treatments. For the Epicor treatment, the butyrate increase was statistically significantly higher than both the cellulose control and the FOS treatment. So let's talk more about short-chain fatty acids. They are produced when beneficial bacteria in your gut break down the fibers in your diet. They are, in fact, an important aspect as to why beneficial bacteria are beneficial. Short-chain fatty acids provide support for a healthy gut by providing energy for gut cells. This helps your gut's immune function and has also been shown to have an anti-inflammatory activity. It has even been shown to have antimicrobial activity, enhancing resistance to pathogenic bacteria such as Salmonella and Clostridium. Lastly, short-chain fatty acids seem to be involved in modulating intestinal motility and sensory motor functions. These functions are involved in stool regularity. So short-chain fatty acid production may be involved in regularity, and Fermentate was shown to affect short-chain fatty acid production. This made scientists wonder if Fermentate could play a role in regularity, and a new human clinical trial was conducted that specifically involved people who suffer from GI discomfort associated with constipation. It was a 6-week, double-blind, placebo-controlled trial on subjects with mild or severe constipation symptoms. The results showed that Fermentate treatment showed improvement in bloating, feelings of fullness, and overall GI discomfort, and that this was statistically significant versus placebo in the mild group. Fermentate treatment also showed improvement over time on surveys addressing the quality of life specifically related to constipation, and surveys that assayed perceived stress. The study went beyond looking at clinical benefits to address possible modes of action. For that, the microbiome was assayed. The microbiome is the world of bacteria that live in your gut, some beneficial and some harmful. And the results of this trial showed that versus the placebo group, those taking Fermentate saw increases over time in beneficial bacterial groups such as bifidobacteria, which immune and digestive benefits were discussed earlier, Bacteroides, which is heavily involved in short-chain fatty acid propionate production, and Akkermansia, which has been negatively correlated with metabolic disorders. In contrast, with Fermentate treatment, there were decreases over time versus placebo in many pathogenic bacterial groups, including Shigella, as well as pathobionts. Pathobionts are bacteria that harm the beneficial bacteria in your gut and thus put it out of balance. Lastly, there were decreases over time versus placebo in the Fermentate group in lactate levels in the feces of the participants. One of the functions of beneficial bacteria in the gut is to break down lactate, converting it into short-chain fatty acids. Thus, this is direct evidence that Fermentate seems to be promoting the beneficial activities that the microbiome can perform. In summary, the extensive human clinical data indicates that a daily 500 milligram dose of this specialized Fermentate, whole food Fermentate, results in a reduction of cold and flu symptoms and a reduction of congestion among allergy sufferers. Thus, Fermentate strengthens the immune response without pro-inflammation. Fermentate has also been shown to have a dual response, activating certain immune factors within 2 hours while working over the extended time needed to give more healthy days. Lastly, emerging science is showing that this specialized Fermentate works in the gut. It supports regularity and digestive comfort and was shown to shift the microbiome in much the same manner as prebiotics, without the bloating seen with the use of prebiotics. So there you have whole food Fermentate, harnessing the metabolites formed in yeast fermentation to provide benefits to both your immunity and your digestive health. Thank you.